Hi, good afternoon, Arena. Hey, sorry about not getting this to you yesterday, but during the uploading process, something happened. After I filmed it and was uploading it, or Loom was processing it, however you want to say it, I was outside getting some yard work done, and we must have had a power blip or short or a surge or something. But anyway, it just failed to process, and so when you'd ask for a second one, I thought, well, I'll just do them both at the same time. So hopefully this works well. Take three on getting this done, but we'll just keep doing whatever we got to do. All right, so let me get a pen here. Looking at this question, the key is that phrase sister chromatids, and I brought some diagrams in here to showcase. This is the karyotype. If you had a cell and you broke down the nucleus and took all the chromosomes out and paired them up based on their function, in other words, what traits they express from both the mother and the father, and put them side by side, you would kind of see they'd be the same shape and same size. And when they're in that form, they're called homologous chromosomes. And I'm going to circle it here, and of course here, and here, and up here. And that just simply means that you've got the chromosome from the father, chromosome from the mother, whatever the offspring is, that express for the same traits. And when I say traits, if we took hair color, for example, this chromosome, if it was for hair, it might express the color, the texture, the width of the hair strands, just anything like that. And so together, the one from the mother, one from the father, the homologous chromosomes. But we're looking for sister chromatids, and sister chromatids you're going to find right here. And what makes them sister chromatids is the fact that we've gone through replication. So we've taken the chromosome from the dad or the mom, whichever one this may be, and duplicated it. And when they're duplicated, now you've got two copies of the same thing side by side, and those are what are called sister chromatids. So if we think about sister chromatids, and we look at the first one sister chromatid is inherited from each parent. No, the sister chromatid is a copy of the chromosome from each parent, so that one's out. Sister chromatids are in every cell, the second choice. We can see down here that once we go through mitosis and those two sister chromatids that are here are separated, now you can see that they're in separate cells. So they're no longer together. And matter of fact, they're only together for just a brief, short period of time. We've gone through the cell cycle, G1, and in the S phase, when they are replicated, then they're side by side. But then when they go through mitosis, and just a few minutes later, they are no longer side by side. They're in separate cells. So that one's out. And if we go down to the one that you did choose, each sister chromatid forms a lobe of a chromosome. Now, I'm not real sure what you were thinking in that process. If we look at kind of a DNA unwound partially, and it's in the chromatin state, these loops, that spiral staircase around the histones, the proteins that help kind of compact them, you may be thinking that loop. You might have also been thinking about, if I draw a straight line to represent a chromosome, there's going to be sections on there in reference to hair that I referenced earlier. Each one of these sections is a gene, and the gene codes for a different trait, maybe color, texture, that kind of stuff. You might have been thinking of that, but either way, no lobes formed on our chromosome. So the one we're looking for is sister chromatids are only present during cell reproduction. That's the choice there. So hopefully that helps. And if we go over to your second question, man, I love Punnett squares. I don't know if you like them or not, but I love working them in class and working with students on those. And you can even take some of the parental traits that they have at home and kind of see how they fared with all that stuff. But anyway, this is kind of a wording situation. I think what happened is you were looking for the percent of the female offspring. Oh, I didn't mean to do that. Let me get my pen back out here. The female offspring to have Duchenne muscular dystrophy disorder. But in essence, that's not what the question is asking you. The question is, what is the probability that a female offspring shown above would have, and I'm going to put a box, would have a recessive allele. So they're just looking for what is the percentage of females that are going to have at least one recessive allele. And of course, as you know, we're using M's. That's the lowercase letter as opposed to the big capital letter. That would be the dominant gene. So a female that would have the recessive trait, and I'm sure you know how to work these, and I'm just going to put the letter M's in here. So this one would be little, this one would be little. I think you were looking for who would have the trait. I could be wrong about that, but you'll know that for certain. And if we just fill this in, that's a big M. M's a tricky letter because it's one you got to make sure you really emphasize the size, or the letters could look the same. This one is going to have an uppercase M, but it's going to have a Y because it's going to be a male, and this one lowercase m, but a male, bringing that down. So these two don't count because they're males. We're looking for a female. And now if we look here, this lady does have, unfortunately, muscular dystrophy. This one does not. The big M saved the day, but they still have the recessive allele. So the reason why they're looking for 100% is because they're not looking for who has the trait. They're looking for who has a recessive allele. So hopefully with this question and with the other question about the sister chromatids, this helps. If not, be sure and let me know, and we can do something else to try to help out. And anytime you have any other questions, and you've been great about asking for help, and I appreciate that. I love making these, love talking this stuff. So until next time, we'll say goodbye.